Hi, welcome to the first TA online session. I'm Johnny, and today we're going to be talking about material from week 2, protein structure, folding, and function. Now when we're talking about protein structure, basically we're talking about what's around this central carbon, which is a hydrogen group, carboxylic acid, carboxy group, and an amine group. The last group is an R group. Now this is the central carbon, and there are 4 groups that we want you to know that are attached to it. The first one is a hydrogen group. We also have a carboxyl group, an amine group, and an R group. Now all amino acids are the same, but being able to know what the structure of an amino acid is helps you to be able to know how it's used and helps you understand other concepts. So I would recommend knowing how to draw this structure. You'll never be asked to do it, but it's helpful to know how. What makes one amino acid different from another type of amino acid is this, the R group. Everything else, which is known as the backbone, basically everything that's not an R group is the backbone, or the backbone molecules. The R group is what gives one amino acid different characteristics from another. So let's talk about the R group. When we're using R groups to describe amino acids, usually we talk about things like if it's acidic or basic, if it's polar or nonpolar. We'll also be talking about if it can be phosphorylated, and then we have some miscellaneous ones that I'll be putting in the special group. Now, a quick review on amino acids. Basically, when one amino acid is linked to another, here's my first amino acid, here's the second, the bond in between them is called a peptide bond. And when we get a bunch of them, we call them a polypeptide chain. That's because it's a chain of many peptide bonds. And yeah, so with the R groups, talking specifically, let's talk about the acidic ones first. So acidic R groups are the ones that will have a negative sign. Now this is a negative sign in the actual R group. Most amino acids, based on the drawing, they all have a negative and a positive sign because they have an acid and a base in the backbone. But when we're talking about whether or not an amino acid is acidic, we're focusing just on the R group. So if in the R group we see a negative sign, that tells us that it's negative. If we showed you a picture of the chemical structure of an amino acid, if you saw a negative sign in the R group, then you know it's acidic. And a quick note on that, to know if it's the R group, you find it whichever one is not the hydrogen, not the carboxylic acid, and not the amine group. It's just the R group. And that's where knowing how to draw one, knowing what it looks like, can come in handy. Now, that's how you can recognize one by its chemical structure, but not always do we show you the chemical structure. So we also want you to know the 2 acidic amino acids by name. The first one is glutamic acid. Then the second one, aspartic acid. Now these are easy because they have the word acid in their name, which tells you they're acidic, but they also both have their own second name. So for glutamic acid, we have glutamate and aspartate. If you see either of these two, we want you to know that they are acidic. Or if you see a negative sign, that's acidic. Now the next group we want to talk about are the basic ones. basic amino acids, they're kind of the opposite of the acidic ones, and so instead of looking for a positive sign, instead of looking for a negative sign, we'll look for a positive sign in the R group. Other than that, we also want you to know them by name. And here's a mnemonic that will help us to remember HAL, or HAL. HAL is the nickname for President Eyring. So think HAL, he is basically an awesome person, an awesome man. Now HAL stands for histidine, arginine, and lysine. Now note that there are other amino acids that start with A and L. Don't get them confused, just keep these three together: histidine, arginine, and lysine. The next group I wanted to talk about is the one that can be phosphorylated. When we're phosphorylating something, we add a phosphate group, which is PO4. These are amino acids that have an OH, a free OH, on the end of their R group. So it'll look something like this. When we have a squiggly line, that just means whatever it's normally attached to. And so this is just the R group. If the R group has an OH here, it can be phosphorylated. So to help us to remember, I want us to remember this. P for phosphorylated, being able to be phosphorylated. S though, serine. And then we have tyrosine and and threonine. These three are able to be phosphorylated. So remember those three. And you can do that by remembering the mnemonic PSTT, or PST. Okay? The last group I wanted to talk about are the special ones, it's kind of the miscellaneous ones, but two special ones that are unique and ones that we want you to remember. The first one is proline. Now, most of the amino acids look just like this. Central carbon with the amine group here, carboxylic acid with the hydrogen and the R group. But for proline, instead of there being just an R group that sticks out, the R group actually is bound back to the nitrogen here. And we'll talk about this later, but that makes it an alpha helix disruptor. When we talk about protein structure, protein folding rather, this will come into play a little bit more. The second one that we want you to know is cysteine. Cystine's R group looks like this. Has a sulfur at the end of the R group. This sulfur, if close to another cysteine, like this, it will be able to make a bond between those two sulfurs, kicking off the hydrogens. And this bond that connects them is called a disulfide bond. This is important because the disulfide bond is the only covalent bond that an amino acid can form with another one. It's the only R group that can form a covalent bond with another. And covalent bonds are strong bonds. So we want you to remember those as well. Because it's a stronger bond, because it's a stronger bond, we will have to add a separate chemical if we want to break down all of the interactions between different amino acids. And that chemical, if you can remember, and we do want you to remember, is beta mercaptoethanol. This chemical breaks disulfide bonds. Now I want to talk to you a little bit about Protein folding. When we're talking about folding, there are different levels of folding. The first one is primary, the primary structure. Primary structure is just the linear sequence of the amino acids. Now, with amino acids, we say that they are written, they have directionality, N to C. That's because when we have a huge chain of them, they run from N to C. This amino acid being the first one that was there, this one added next, and so forth. Now, the linear sequence is just the order that they're in. So if I have an arginine with a leucine, and a serine, and a valine, that is the primary structure, just the order of these amino acids. The secondary structure, though, secondary structure is when we start having interaction, and this is hydrogen bonding between the backbone atoms. Backbone atoms, if you'll recall it, are the NCC, anything that's not an R group. So if we have a bunch of these together, they'll naturally form bonds with each other. This that I've drawn up is a crude sketch of an alpha helix. There's also beta sheets that we want you to know. These are the two general forms of the secondary structure. After that, we have the tertiary structure. Now this is any interaction between different R groups. So before we talked about the backbone, and now we increase in complexity where it's in the R groups now. And this can be anything from hydrophobic hydrophilic, interactions. It can be ionic. So if there's an acidic amino acid close to a basic one, they'll form plus and minuses will attract and form an ionic bond. We also have hydrogen bonding within the R groups. We also have cysteine, right, making disulfide bonds. These determine the tertiary structure. The last one we wanted to talk about is quaternary. Quaternary structure is similar to tertiary, where they're the same, these types of interactions, but now it's between separate subunits. So if I have an amino acid, so what I mean by separate subunits, I mean separate chains, okay? Separate polypeptide chains. So if I had here's one polypeptide chain, and here's a neighboring one that is bound to with another one here, then I would say this has 3 subunits. That's how we describe quaternary structure. It has 3. We measure it by the number of subunits it has. Now back in here, when I talked about hydrophobic and hydrophilic, this refers to whether or not the R group is polar or nonpolar. If the R group is polar, which is similar to water, here's a water molecule, right? Oxygen with 2 hydrogens bound to it. Oxygen likes electrons, it's electronegative, so this area of the water molecule is negative, whereas this one is positive. And because of this polarity, it's polar. And molecules that are also polar, and when we mean polar, we mean charges that aren't balanced within the whole molecule. When molecules are polar, they like water. They like to be around water. Now, molecules that are not polar don't like water. So those are described as hydrophobic. So nonpolar, hydrophobic, and polar is hydrophilic. Sorry, this looks really bad, but yes. So to summarize that, to restate that, molecules that are polar are hydrophilic, they like water because water is also polar. Molecules that are nonpolar don't like water because they're different. And as far as a protein goes, if there are areas where there are a lot of amino acids that are hydrophobic, they will tend to be on the inside. And when the protein wraps together, they'll try to be on the inside to be as far away from the water as they can. Hydrophilic ones will tend to be on the outside, where they want to be next to the water. Now that we've talked about structure, protein structure, we need to also talk about function. Function is really important because all of the things that go on in our cells, they require enzymes. And the function of those enzymes is very specific. And the function, sorry, I'm going to say it this way, the structure of that amino acid chain or the enzyme is what determines its function. If the shape changes, the function is either decreases or is taken away completely. And when we call a change in shape, we call a change in shape denaturing. So when an enzyme or when a protein denatures, the shape has been changed. And that can either be breaking down of all the bonds that we talked about before, or any other type of change in shape. And so usually, heat, temperature will break the weaker bonds, like hydrogen bonds. Also, if it's acidic, if we put them with acid, those bonds will break and the protein will denature, as well as other chemicals like the beta mercaptanethanol, and so on. Protein structure is important, once again, because the structure determines its function. If we change the amino acid through something like a mutation, or if we change the shape by adding or doing something with the environment of that amino acid, if it changes shape, then it won't be able to do its job anymore. Okay, now I forgot to mention about proline, one of the special amino acids. Now if I go back to the alpha helix, remember proline is an alpha helix disruptor. So because proline looks like this, notice that because secondary structure is determined by hydrogen bonding in the backbone, that's the oxygen of one amino acid interacting and making a hydrogen bond with the amine of the other. And actually the hydrogen of the other. But because notice that proline's R group is bound back to the nitrogen. Because of this, it's not able to make a hydrogen bond with another molecule. And because of that, it disrupts the alpha helix. Also, a few quick facts about proteins. Some quick facts I'll draw here. There are about 110 Daltons, that's how big they are. And they absorb the 280 wavelength. This is nanometers. Okay, facts about proteins. In the next video, we'll talk about the different methods that we can use to purify proteins, as well as a few of the experiments that are in our packet. But before I go on, I want to just quickly recap the things that we've talked about. If you'd like to, go ahead and skip to the next video. Okay, so just running quickly through, I'm going to write with red this time. Know the chemical structure of an amino acid, as well as the 4 groups: a hydrogen group, a carboxy group, an R group, and an amine group. What makes one type of amino acid different from another type is this R group, and these are the different characteristics of an R group. Once again, proteins go in the direction N to C. Not only are they counted that way, but they're also written that way when they're translated. Acidic amino acids are the ones that have negative signs. So if you see an amino acid that has a negative sign, it's acidic. Also know these 2 by name. Basic ones are the ones that have a positive sign. Know these 3 by name. The ones that can be phosphorylated are the ones that have OH in their R group. Know these 3 by name. Also, proline, an alpha helix destructor, because its R group is bound back to the backbone. And cysteine can make a covalent bond with another cysteine. That's the only amino acid that can make a covalent bond. That covalent bond is called a disulfide bond. To break that bond, we add a chemical here, beta-mercaptoethanol. Now, there are different levels of structure. The first one is the linear sequence, just basically the order that they're in. The next one is hydrogen bonding in the backbone. That's secondary structure. Tertiary structure is any interactions between the R group. Quaternary, between different subunits. Last thing to remember is with the function. The structure is what determines the function. And if we change the structure, we will change how well it can do its function.